the pathways, how to utilize and how to form the amino acids. And then in the second part, we will review the derivatives of amino acids that we produce. So first, we start with the utilization of carbon skeleton. Yesterday we did the utilization of ammonia. So now what we have is carbon, hydrogen, and oxygen inside the carbon skeleton. First, what's good to say is that each amino acid has its own individual pathway. Some pathways are long, some pathways are short, but for each you can define some individual enzymes. As the result, you will produce these 7 intermediates for all amino acids. First, acetyl-CoA and acetoacetyl-CoA, as you know, are precursors or are intermediates of metabolism of fatty acids. That means they can give birth to ketone bodies, and therefore, if amino acid gives one of these 2 intermediates, we call it ketogenic amino acid. Then we have 5 other intermediates that can be produced from amino acids. In majority, they are the intermediates of Krebs cycle or pyruvate. And since you know that all intermediates of Krebs cycle and pyruvate also can be converted to glucose, we call these amino acids glucogenic amino acids. If we look at the list, the majority of amino acids are glucogenic, only 2 are ketogenic. Ketogenic are only those 2 that begin with L, thus you can remember it, so it's lysine and leucine. Then as you see, the biggest group is the glucogenic, and then we have one special group for 4 amino acids, for all aromatic amino acids, and amino acid leucine, that gives you 2 intermediates. One from this group and one from this group. And therefore we call them keto and glucogenic amino acids. Of course, in the test you can have variation. So we can ask you not only on the distribution, that means whether the amino acid is glucogenic or ketogenic, but we can ask you whether from this amino acid we can form glucose. So that means that from all these amino acids you can form glucose. And of course we can ask you, for example, if you can from these amino acids produce fatty acids. The answer is also yes, since from pyruvate you can simply produce acetyl-CoA, and therefore you can convert these amino acids after quite long, but after some time, yes, to the fatty acids. That means that ketogenic and glucogenic means what intermediates do you produce. But then you can say that from all glucogenic you can produce the fatty acids. Do you understand this difference, what you can meet in the statements and in the test?
So all of them can produce fatty acids?
What?
All of them can produce fatty acids?
Yes. All can give birth to fatty acids. And as you know, if you have excess of amino acids, you finally convert it to fat or you burn it. That's the second possibility. But here we do not speak about the burning. The burning is after all, and if you continue with the metabolism, you will incorporate them inside the metabolism and you will burn them. But the keto and glucogenic means whether one of the intermediate will be from this group or from this group. So it's only the artificial division. If you look into your books, you can see quite big chapter about this. That means how different amino acids are through sometimes simple, sometimes more complete pathway incorporated into the energy metabolism. That means how do you produce from the amino acid one of those 7 intermediates. We are not going to review it in big detail today, since the importance for you is some, but in the clinical practice, really the detailed knowledge of degradation of amino acid carbon skeleton is limited to very focused group of doctors. So if you want to do pediatrician focus on the inborn errors of metabolism, you have to know it. But you can ask yourself how many of you will be doing this. That means that we decided that we do not expect you to know in detail catabolism of all amino acids, but only some of them that we will show here today to you. So you can look at it in the chapter. It's recommended for you at least one time to read it, so to have some idea how the whole system works. But we will not ask you at each enzyme, for example, in the degradation of tryptophan. That's a very big, long pathway, and the amount of energy that you must invest into this, we think that you can utilize in much better way. Only for you, if you see, really the pathways can be highly variable. If you look at the branch chain amino acid, one is purely ketogenic. Yes, that leucine. Valine is purely glucogenic, and isoleucine is keto and glucogenic. So that means that even if you have these amino acids in the same group, according to the structure, according to the nomenclature, they will give you different intermediates. So really there are some rules, but sometimes not so strict rules, and the fate can be different. Here you have some simplification with no enzymes, but only with the name of the intermediate and the abbreviation of the amino acid, plus you have here the color, what group of amino acid does it represent. So you can, of course, learn it if you want, but it won't be the main topic of the test, yes? You can have there, for example, some question on the defect, but then we are asking you more on the general understanding. What does it mean if you have defect somewhere? That means that you will accumulate the substrate and you will be missing the product. Yes, so that can be the question on. What you should know, and this is the minimal thing that we really expect you to know, and this is for us the basic knowledge, are these amino acids. Where you must know how they are degraded. First 5, if we take this together, you already know. They have very basic degradation. If we start with glutamine, you know that glutamine is converted to glutamate by glutaminase, and then glutamate to alpha-ketoglutarate by transamination or oxidative deamination. So this pathway you must know, and that's for us the basic knowledge. The same you must know for asparagine and aspartate. The same thing, asparagine is hydrolyzed to aspartate, and then aspartate is transaminated to oxalacetate by aspartate aminotransferase. And third here is alanine. Alanine is transaminated by alanine aminotransferase, producing pyruvate, so the simple transamination. And here you see that for some amino acids is the degradation pathway really, really very simple. That we want you to know the lysine and leucine, that they are ketogenic, and that means that they give you the acetyl-CoA and acetoacetyl-CoA. And the second new thing is that those, what we can call small amino acids, that means glycine, serine, and cysteine, are all glucogenic, and they are converted to pyruvate. We will also use this for their synthesis. And other thing that you should know to some small detail is the oxidation of branch chain amino acid. As you know, we mentioned to you repeatedly that branched-chain amino acids are, in the first reaction, metabolized in the skeletal muscle. You should know first two steps of the degradation of branched-chain amino acids, since these have some clinical significance for many of you. The first reaction for all branched-chain amino acids is the same. It's the special transaminase in skeletal muscle that converts this amino group. Here you have leucine, valine, and isoleucine. It converts it to the keto group. These three molecules we call alpha-keto analogues of branched-chain amino acids. And you will meet them if you will do nutritional interventions in some person, for example, in person that have liver problem. Therefore, we want you to know the branch chain or alpha-keto analogues of branch-chain amino acids, since you will this term meet quite frequently. Then this intermediate comes into the liver, and in the liver you do the second reaction. That's the oxidation plus decarboxylation. So we call it oxidative decarboxylation, and the product will be acetyl-CoA. If you look at it, you already know this reaction, for it's the very similar principle as the reaction between pyruvate and acetyl coenzyme A. So here you have keto group, here you have carboxy group. You decarboxylate, that means you remove this carboxy group. You oxidize this keto group to carboxy group, and at the same time you bind coenzyme A to it. So here you produce acyls-CoA derived from the alpha-keto analogues of branched-chain amino acids. And this enzyme, that's the enzyme that is defective in one disease that we call maple syrup urine disease. And that's one of the diseases that you will speak with genetics about.
Can you repeat?
Maple syrup urine disease. Yes. And why it's maple syrup? Since the urine has the smell as the maple syrup. Yes. What precisely will happen, you will speak later. Yes. But since you will meet this enzyme and you will read about it quite often in the books, since it's causing this disease, and that's one representative of the amino acid diseases, you should know it. You should know at least this step. The further steps, you do not have to know in detail. You do not have to know them. They will lead to other intermediates. But what you should know for branched-chain amino acids are these two. The transamination at the level of kidneys, at the level of skeletal muscle, and the oxidative decarboxylation in the liver. And the fact of this enzyme leads to the maple syrup urine disease. So, any other question?
What is the name of the one? The S-CoA and the other?
This carbon dioxide.
No, the one name. In the isovalent way and the other.
This?
No, the one down. Yeah.
Isobutyryl-CoA.
Yeah, the one name between the CoA and the other molecules.
This is the sulfur. Or right now, I do not know. What do you mean?
Like, isn't it like the thioester bond or something?
Yes, this is the thiester bond. That's the same bond as you have in the acetyl-CoA derivatives in general. So that's it. That was the utilization of carbon skeleton. So you should know this list, and you should know the basic steps. You can look at the other things, but they won't be so condensed inside the test. Second part is the utilization, is the formation of non-essential amino acids. So before we look at them, first important information is that we cannot produce essential amino acids. So in the test you can meet variations of the questions, for example, that valine is produced from pyruvate. We do not expect you to know it, since you know that it's essential, therefore we cannot produce it. Yes, so this is only the questions whether you understand what does it mean if compound is essential. Yes, essential compounds we cannot produce, therefore they have no precursor in our body. So if you meet this kind of statement, it's incorrect. Then for those, yes? No. Then for those that are not essential, here you have the list, how do we produce them. So if we look at those that were at the previous list at the top, that means alanine, glutamate, glutamine, aspartate, asparagine. You see that they are produced quite simple by the transamination from pyruvate, alpha-ketoglutarate, oxalacetate, or for glutamate you can reverse the glutamate dehydrogenase reaction. So that's quite simple. New thing is that you should know that proline is produced by alpha-ketoglutarate, and then those three small, how we call them, amino acids are, that means serine, cysteine, and glycine, are produced from intermediate of glycolysis from 3-phosphoglycerate. As you know, in the glycolysis you had 1,3-bisphosphoglycerate that was converted to 3-phosphoglycerate, and then you have 2-phosphoglycerate. And this 3-phosphoglycerate serves as the precursor for serine, cysteine, and glycine. You do not have to know in detail how it's done, but you should know that these three non-essential amino acids are produced from the precursor from the intermediate of glycolysis. That means the same that glycerol is produced from dihydroxyacetone phosphate. The one, or the last, that was here, that's the production of tyrosine. Tyrosine is for our body not an essential amino acid if you have phenylalanine. Since we have very simple reaction how to convert phenylalanine to tyrosine. You know that the difference between these two amino acids is only this one hydroxy group. And therefore it's not surprising that the enzyme will be called hydroxylase, and the substrate is phenylalanine, therefore phenylalanine hydroxylase. This enzyme needs to have a cofactor. The cofactor is called Tetrahydrobiopterin. That means that it has 4 hydrogen, and 2 of them it will incorporate into the reaction, plus it will use the oxygen molecule. 1 oxygen will be incorporated into tyrosine, and 1 will be incorporated into the water. This is one way how hydroxylases do the reaction. They take molecular oxygen, then some reduced compound, incorporate 1 oxygen inside the compound of interest, and forming water. That means that you must have second enzyme that regenerates the dihydrobiopterin back to the tetrahydrobiopterin, and that's the reductase. So directly here you do not consume reduced cofactor, you use it in the other enzyme. Why do we want you to know this enzyme? The phenylalanine hydroxylase is due to the fact that its enzyme defect stands behind other quite known disease, and the absence or reduced activity of phenylalanine hydroxylase leads to phenylketonuria. Phenylketonuria is screened in all our newborns, and the reason for this is that if you have a defective enzyme, the phenylalanine cannot be converted to tyrosine, and it will look for alternative pathways how it can be metabolized. One pathway you have here, that's the production of phenylpyruvate from phenylalanine. The reaction is the transamination, and you simply take this amino acid to alpha keto acid, and therefore here you have the keto group. Subsequently, this keto group can be reduced to phenylactate, and then you can have the phenylacetate. These compounds you assess in the urine, and you can know that when these compounds are produced in higher quantities, that there must be the block of the phenylalanine hydroxylase. And one other alternative pathway, an alternative product, is phenylethylamine. That means that, as you see here, you will miss the carboxy group. So it's the amine derivative from phenylalanine, and this compound is toxic to your brain. And therefore, if the level of phenylalanine is high and it cannot be converted to tyrosine, you will produce an increased number, this toxic compound, and it will lead to irreversible brain damage. And of course, especially susceptible to this damage is the developing brain during the newborn period and during the childhood. Therefore, if you want to prevent the development of this brain damage, you must do the screening at the very beginning of the life, and therefore all children or newborns are screened to it. How you could treat this disease? Could we cure it, or what we can do with it?
We can. Restricted diet.
Yes, the thing what we do is the restrict of phenylalanine in the diet. Then the phenylalanine does not accumulate. Therefore, typically you do not have the absolute absence, but you have only the reduced activity, is able to metabolize it. Therefore, you do not have the accumulation of these toxic intermediates to the toxic levels, and the brain can develop. Of course, that these diets you must have, or this special artificial diet, you must eat for quite a long time, and unfortunately the diet is not very tasty. Yes, but it's the quite good thing what you can do if you have enzyme defect. Then if you will not give so many substrate, then the reduced activity of the enzyme can metabolize it, and you do not have no accumulation. Very similar thing you will see in one of the detective stories. One of the group will see how one enzyme defect will lead you also to the modification of the diet of the patient. And it's the same principle as the phenylketonuria. So any question to this?
For the reaction, is it necessary to know the structure of the coenzyme, or is it just necessary?
Not, you must know this. And that you should know that there is the Tetrahydrobiopterin, but you do not have to be able to draw it. You can have there this structure, so this whole picture, so that means you must be able to identify phenylalanine and tyrosine, and that you know that this is the reduced form, since here you have more hydrogen, as you see inside. Here you have, these hydrogens are here missing. So this is the form, the tetrahydro. This is only the dihydro form.
I have a question about phenylate. How it's transformed with phenyl acetate?
You mean how this, by decarboxylation you do it? Oxidative decarboxylation.
Okay.
That was the story that's also in the books, sometimes in quite big detail. Yes, both the formation of non-essential and the utilization of carbon skeleton. So what we expect you to know is this basic overview, and you must know it in the detail. Yes, this is the basic knowledge for us. What we will do now are the derivatives of the amino acids. First, what we start with are the derivatives coming from the decarboxylation of amino acids. As you know, if you decarboxylate amino acid, you will keep there the amino group, and you form molecules that we call biogenic amines. These biogenic amines play a very important role in the whole body metabolism. And as you will see, especially they are important for function of our central nervous system. If we start with tyrosine. Tyrosine is an amino acid that will be hydroxylated, subsequently decarboxylated, and it gives birth to a group of compounds that we call catecholamines. Catecholamines, that means noradrenaline, adrenaline, and dopamine. As you know, adrenaline is a very important stress hormone, and it's one of those control-regulatory hormones. Noradrenaline plus dopamine, they are important neurotransmitters. For example, dopamine, the lack of dopamine leads to the development of Parkinson's disease. And dopamine also has the role in development of addiction. So for example, cocaine is increasing the dopamine concentration inside the synapse. Second example is tryptophan. Tryptophan is converted to compound called serotonin. The systemic name is 5-hydroxytryptamine. So also here you see the amine. That means compound derived from amino acid by decarboxylation. As you know, serotonin is other example of neurotransmitters, and it's important for your mood. If you have patient with depression, you give him drugs that block the reuptake of serotonin from the synaptic cleft, returning the mood to the standard setting. This is more pharmacotherapy. You will review it in the next course with Dr. Trnka. That are called SSRI, selective serotonin reuptake inhibitors, and they lead to increase of serotonin in the synapse. But you will review this. How does it precisely work? Yes. Right now, only know serotonin neurotransmitter. Histamine is derived from amino acid histidine, also by decarboxylation. And what's the role of histamine in your body? Yes, allergic reaction is something far, but before it we have normal immune response. Of course, when it's too strong, then we have the allergy. And other thing, it's important for the regulation of sleep-wake cycle. So if you had old antihistaminic drugs, you feel drowsiness after them, since you block the effect of histamine as a neurotransmitter. Other compound that can be decarboxylated is serine. Serine, by decarboxylation, gives you ethanolamine. And ethanolamine, as you know, is the constituent for the phospholipids. When you do a triple methylation of nitrogen in ethanolamine, you will produce choline, and choline is other part of the phospholipids. And then if you add choline plus acetyl-CoA, you produce acetylcholine, and acetylcholine is other neurotransmitter that you need to all macroscopic movements, since it's the neurotransmitter at the neuromuscular junction. And also, acetylcholine is a neurotransmitter in your brain, and the lack of cholinergic neurotransmission leads to the development of Alzheimer's disease. And at the very beginning, you had the amino acid. Then if we close the story for neurotransmitters, glutamate is decarboxylated to gamma-aminobutyric acid, abbreviated as GABA, and this is the most common inhibitory neurotransmitter of our brain. Glutamate is the most common excitatory neurotransmitter of our brain, and GABA is the most common inhibitory one. So you see that the difference for your brain in inhibition and excitation, you can say it's one decarboxylation. So very, very small difference in the shape of the molecule. And if you return here, cysteine is decarboxylated to cysteamine, aspartate to beta-alanine, and these two molecules together are some precursors for the coenzyme A molecule. That means the molecule that you need for the metabolism of, for example, fatty acids, or for the metabolism for the cholesterol synthesis. So you see that biogenic amines are really very important group of compounds. You see how deeply we have them in the metabolism, and also how big number of neurotransmitters are derived from amino acids or either are directly amino acids. So any question to this? If not, then we will look on the second. Now it's not a biogenic amine, but it's the small molecule called nitric oxide. Nitric oxide is produced from amino acid arginine that's converted by enzyme called NO synthase to citrulline. And as you know from previous lecture, the difference between citrulline and arginine is quite small, only here. And if you combine the enzyme with oxygen and reduce NADPH, you here have nitric oxide produced. Nitric oxide is a molecule that stands behind many important processes in our body. If you will look at it, it was also 1 year, a molecule of a year. So it's very big award for the molecule to be a molecule of year. And it was since discoveries that this quite small molecule that was thought to be only a radical plays, we can mention these three main functions. First is that NO synthase is inside your brain and it modulates the neurotransmission inside the brain. Second is, since it's a radical, it can be used to destroy organisms that are foreign to your body. And nitric oxide is representative of reactive nitrogen species that are together with reactive oxygen species produced in your professional macrophages or professional phagocytes, for example macrophages, to destroy the bacteria. So nitric oxide is produced here to destroy the pathogens. And the last is knowledge that endothelial cells produce nitric oxide as one of the main vasodilatory compound. That means the compound that increase the diameter of your blood vessels and therefore lowering the blood pressure. And you must have a balance between the production of vasodilators and vasoconstrictors to keep your normal blood pressure. And nitric oxide was found to be really crucial in the process of keeping a normal blood pressure at the level of endothelial cells. These things have some correlations for you. If you read all books, you can read them that people that have angina pectoris, that means narrowing of coronary arteries in your heart, if they have the attack of angina pectoris, they take the nitrates. The explanation is quite simple. Nitrates will generate nitric oxide, therefore causing vasodilation, and therefore you will have for some time improved vascularization of the heart muscle. Nowadays, this is not prescribed so routinely. We use in the medicine some generators of nitric oxide, but it's really not used as the first-line treatment. We have better drugs, but still the mechanism that's here you can find in the medicine. Second, where you will find not the positive effect of nitric oxide, but negative effect, is in the patients that have severe infection. That we call a sepsis, that means the generalized infection. And in these patients, one of your problem will be that they typically have very low blood pressure. And we call this a septic shock. That means the situation when the circulation is not able to keep adequate blood pressure to enable the nutrition of your tissues. And the one reason that stands behind this is the production of nitric oxide. So when you have the pathogens, you want to destroy them by the immune cells. But unfortunately, this nitric oxide will also modulate the vascularization. And if you have overproduction of nitric oxide, the result will be hypertension. And when you go to the hospital, to the intensive care units, you will see that sometimes for the doctors it's almost impossible, even when they use very high doses of vasopressors, that mean drugs that increase the blood pressure, to keep some normal blood pressure in these patients. Simply the effect of nitric oxide is so strong that it's almost impossible to overwhelm it if you have really severe septic shock. So, do you understand this? Third example of derivatives of amino acids. If you open a chapter about thyroid hormones, you will read that they are derivatives of amino acid tyrosine. But they were not mentioned in the part about biogenic amides. And that's due to the fact that they are derivatives and as you see, they contain some part of the molecule inside, but they are not decarboxylated. We will speak in detail about the synthesis the next course. So now only briefly, for thyroid hormones, you must have 2 molecules of tyrosine and iodine. As you see, here you do not have the free tyrosine, but it's bound in a very big precursor molecule that we call thyroglobulin. So you have big precursor molecule that's rich in tyrosines, and in the first step you will incorporate the iodine to it. Then we gain monoiod tyrosine, and if you add 2 iodines, you have the iodotyrosine. Not. Only right now to explain you what is it. You should know that it's tyrosine, that you need 2 molecules, and that you need iodines. But if you do not have to memorize it only in this step, I now briefly want to show it to you how it's done. So now at the level of monoiodotyrosine or diiodotyrosine, you have the prepared amino acid, and as you see, the final molecule consists of 2 aromatic rings. So in the last step you will do the coupling. That means that you will join these 2 amino acids together, forming one it's 3 iodines and one it's 4 iodine. So thyroid hormones are derivatives of tyrosine. They are iodinated, but they are not decarboxylated. So that means that they do not belong to the biogenic amine group. They are produced by the coupling of tyrosine molecules together, and the molecules were in previous step iodinated. And therefore here you can have iodine in the final molecule. The final hormones, the names you should know, that's thyroxine and triiodothyronine. As these intermediates you will learn in the next course. Last before the break, to close the tyrosine gives you one pigment also. If you do the oxidation and polymeration, it's quite long process, so we will take only the result. The result of oxidation and polymeration of tyrosine is the melanin. Melanin in our body typically exists in 2 forms, or in 2 locations. One, it's the melanin that you have in your skin and in your eye, and this is produced from the melanocytes. As you know, these cells, they typically produce, when they are in rich quantity at one place, quite high amount of the brown-black pigment, and that's the melanin. Second group is the melanin that you can find in your brain. So we call it neuromelanin. And if you should name one place, that's place that we call substantia nigra. And one part of the substantia nigra will be defective in the Parkinson's disease. So you can remember that tyrosine gives you dopamine as the biogenic amine, plus tyrosine gives you melanin, and therefore something what's black in your brain, that means substantia nigra, must have something to do with dopamine, and altogether it's coupled together in the Parkinson's disease. So, question to this.
What does it have to do with dopamine?
Since lack of dopamine in basal ganglia will lead to the Parkinson's disease. How it's done, that's behind the first year, that you will learn in the third and in the fourth, yes? But it can slightly help you that you know that melanin, tyrosine, dopamine, tyrosine, therefore it must be at the same place. So, if no question, then we will take break till 24. Now we will look at derivative of amino acid methionine. You, I think, already know the compound abbreviated as SAM, that means S-adenosyl methionine, since it's the compound that is used as the enzyme cofactor in reactions where you want to add some single carbon on. Other cofactors that you know are the derivatives of tetrahydrofolate, for example, methyl tetrahydrofolate. So S-adenosyl methionine is produced by quite simple reaction. You take methionine plus ATP, you remove the phosphate, and here you have the methionine bound to adenosyl through the sulfur. And here you see this methyl that can be used for the methylation. For example, this S-adenosyl methionine is used for the synthesis of adrenaline from noradrenaline, so inside the synthesis of catecholamines, or in the synthesis of choline. That means the methylation of phenylamine to choline. So those reactions that we already mentioned today. Any question to this? You should be able to identify what's S-adenosyl methionine. We do not expect you to be able to draw it in whole, yes? But when you see this, you should be able to identify methionine plus adenosyl, that you should be able to do. Other derivative of amino acid is the compound that you know from practical trainings, and that's creatine. Creatine is produced from two amino acids. It's produced from glycine and from part of the molecule of arginine. So if you take this part of arginine, you produce the ornithine, so other amino acid that you know from the urea cycle, and you know how in close relation these two amino acids are. So you take these, one carbon and two nitrogen atoms, you combine them with the glycine, here you gain one intermediate, and in the next step you use the S-adenosyl methionine to methylate the intermediate. Here you add a new methyl, and you produce the creatinine. So creatinine is produced from two amino acids, from arginine and from glycine. As you know, in your skeletal muscle, these compounds serve as the energy reserve in the form of phosphocreatine. It's able to incorporate the phosphate to the molecule of ADP, thus regenerating the ATP molecule. And after some time, by a non-enzymatic transformation, it produces a cycle that's creatinine, and creatinine is the waste product that will be excreted from your body by the kidneys. So any question to this? So that was about the amino acid derivatives. You should be able to do it or to write them in the list when we will ask you, for example, on biogenic amines, and you will give, for example, 2, 3, 4 examples, or that we will ask you what derivatives of, for example, glycine, or what derivatives of tyrosine do you know? One thing I will add to you, you will meet, of course, many other derivatives. For example, one that could be quite useful for you is that the glycine, and you can read quite often about it in the book, is the derivative or is the precursor for the synthesis of heme. That means that the part of the cytochromes, the part of the hemoglobin, the heme part is produced from glycine plus from the succinyl-CoA from the Krebs cycle. So that can be one question. Plus glycine work as the precursor for purines. about both these pathways, synthesis of heme and synthesis of purine, you will have separate lectures in the next course and then in the 6th course. So you do not have to know them in detail, but it's useful to know that glycine is the precursor of heme plus purines. That means for both. And how precisely it's used, you do not have to know. There's only this knowledge that glycine not only for creatine, but also for these quite big molecules. So now if we go to the organ specifics. Before we come to organs, we will look at your blood. You know that in your blood you can quite easily transfer polar compounds. Amino acids are in big extent polar compounds, so they can be measured in your blood. The total concentration of amino acid, if you take it as a sum, is between 2.3 to 4 millimoles. So it's slightly lower than the concentration of glucose. You should know concentration of 2 amino acids. First is glutamine. As you know, the main transfer form of ammonia in your body. That reaches the concentration around 0.6 millimoles. And then alanine, that's the second important transfer form, and that's 0.3 millimoles. And then you should know concentration of 3 waste products that are typically transferred from your body to the kidneys for the excretion. And that's ammonia, as you know, has the lowest concentration since it's toxic. So the maximal value should be around 35-40 micromoles. Then you have creatinine that has higher concentration. The concentration is different for males and females. Males have typically slightly higher since they have more or higher amount of muscle mass. Women typically have slightly lower. But there can be very big intra-individual differences. If you have the obese male and women with high physical activity, their values can be very similar. So creatinine levels you must extrapolate to the person you are sitting from. So if you have cachectic old lady, these values are too high for her. If you have young ice hockey player, these values can be too low for him. So really here you must correspond them with the person you are sitting for. Urea has the highest concentration, is non-toxic compound, and you can have them typically between 2 and 8. But if you have, for example, concentration 20, it's not toxic for you, it's only marker for you that typically there is some problem with the kidneys, since urea should be excreted in the kidneys from the body. If we now come to liver, that means some real organ. As you heard during these 2 hours, inside the liver you do many important reactions for the amino acid metabolism. So if we slightly review them and repeat them, you typically remove the amino group from amino acids inside. Then subsequently, when you have the free ammonia, you detoxify this ammonia in liver cells to urea. That means that liver cells are the place where you remove the amino nitrogen and subsequently detoxify it, and then liver cells will excrete the urea into the bloodstream and kidneys will excrete them from the body. Also in liver, you have quite intensive metabolism of the carbon skeleton. That means that if you have, for example, the excess of amino acids in the diet, inside the liver, you can convert these amino acids, their carbon skeleton, into fatty acids, into triacylglycerols. On the other hand, if you are in starvation period, you do the proteolysis of your own proteins from skeletal muscle, for example. Inside the liver, you will transform the carbon skeleton to the glucose molecules in the process of gluconeogenesis. Or simply you can burn the carbon skeleton inside the liver when the liver cells are aiming for the energy. And the last function that's important for the amino acid metabolism, liver cells, that's the important place for the production of non-essential amino acids. As you saw, the production of non-essential amino acids, it's sometimes only the reversion of the degradation, typically the transaminases. So any question to this, to the liver? Intestine. Inside the intestine, you come into very close contact between the amino acids in the diet, and the enterocytes must decide whether, and when yes, then how they will modify the amino acids from the diet from those that they will excrete to or secrete to your body. For some amino acids, they really modify the composition of the amino acids from the proteins in the diet and the composition of the amino acids that they secrete into the bloodstream. So enterocytes actively modify the composition of the amino acids, and here you have some examples. Typically, they secrete higher amount of proline and citrulline, and on the other hand, they consume glutamate and glutamine. So they secrete lower amount of these two amino acids. Glutamine altogether has very unique position for enterocytes. You were heard during this year that glutamine is a very good source of energy for rapidly dividing cells. And as you know, enterocytes are quite rapidly dividing, half-life 3 to 5 days. Other example, that's for example some of our immune cells that also must be replaced quite quickly. The question is why glutamine is so good source of energy for these cells. And there are two reasons for this. First, glutamine can be simply converted to a component of Krebs cycle to alpha-ketoglutarate. That means you have very quick source of energy, and rapidly dividing cells must have enough energy for the whole process. And second, as you will learn in the next course, the amino group of amino group of glutamine is important for the production of nucleic bases. And rapidly dividing cells, of course, must have adequate production of nucleic bases. That means in glutamine you have at one molecule two good things for rapidly dividing tissues. Substrate for the production of nucleic bases, and carbon skeleton of glutamate can be simply utilized as an energy source. The question is, what happens if you have no food in your GI tract? Therefore, you have no glutamine inside GI tract, and the question is whether the enterocytes are not starving. To some extent, they starve, but this is limited by the fact that skeletal muscle cells during starvation work as the main source of glutamine for your rapidly dividing tissues. And the skeletal muscles will, on the other hand, produce more glutamine and alanine than they have inside their proteins. And they do this actively since they will feed these rapidly dividing cells. And this whole process we call the glutaminolysis. It means the interaction between organs. Here, intestine work as the consumer of the glutamine that's produced inside the skeletal muscle cells during starvation or during some metabolic stress. So, do you understand it? Almost, so what not?
If I go through it one more time, I'll understand.
Skeletal muscle. So, first we can say that skeletal muscle is the biggest storage of proteins. As you know, these proteins have inside skeletal muscle some function, but if you must degrade some proteins, then the loss of the function in skeletal muscle is not so severe. So you will use it as a primary source of new amino acids. This I already told to you. So skeletal muscle cells, the same as enterocytes, actively change the spectrum of amino acids that they release from their molecules or from their bodies. That means, as you know, branched chain amino acids will be transaminated at the level of skeletal muscle, and into the bloodstream you release the alpha keto analogues of branched chain amino acids, plus the nitrogen from this will be incorporated into the alanine and into the glutamate and glutamine. And these are sent to the rest of the body. Glutamine can be used in the rapidly dividing cells. And as you know, alanine typically enters liver cells. In liver cells, it's transaminated by ALT. Amino nitrogen is utilized in the urea synthesis, and pyruvate that's formed is converted to the glucose. You produce the glucose molecule that can enter the bloodstream and can be cached in the tissues that need it, or if we want to close the cycle, it will return to the skeletal muscle cells. And as you know, if you compare this with the Krebs cycle, this cycle is less energetically effective, since here you produce 2 ATPs, here you consume 6 ATPs, that's the Krebs cycle, but here you consume 4 other macroenergic bonds in ATP molecule. So from this perspective, it's less effective. On the other hand, it's more effective in the perspective that here you gain the alpha keto acid that can be metabolized. And this comes from the amino acid from the skeletal muscle reserves. So, do you understand skeletal muscle and the Krebs cycle? And then the last, that are the kidneys. In the kidneys, you, as a basic main role, you do the excretion of nitrogen waste compounds. That means in kidneys you excrete urea, you excrete creatinine, you excrete free ammonia, and from purines you, for example, produce uric acid, and that you excrete also in the kidneys. That means that if you have any kind of kidney failure, these compounds will arise in your bloodstream, will arise their plasma concentration. Some of them are not non-toxic, so you can have, for example, 500 creatinine, and it's non-toxic. Some of them, unfortunately, are toxic. Yes, so urea plus creatinine, they are typically markers of the kidney function, and you know that you should be afraid whether other waste compound is not also increasing and therefore endangering your life. If we return to the fact that we mentioned tomorrow, that was the fact that urea is the predominant excretion form of nitrogen, and that ammonia is only the second form. Sometimes they can be equal, and as we mentioned, it could be, for example, in the episodes of acidosis. since during acidosis you decrease the velocity of the urea cycle. And in that moment, your kidney cells will actively take the glutamine, convert it by glutaminase to glutamate, and then do the oxidative deamination to alpha-ketoglutarate. And ammonia that is released from these reactions will be excreted directly into the urine. And in that form, the ammonia typically catch the hydrogen proton, and therefore you will, by this mechanism, lose the protons from your body. And that's the thing what you want to do if you have acidosis. So inside the tubular cells in kidneys, you share some enzymes in hepatocytes, and their concentration and their activities are different in different occasions during your life. So if you have periods of low pH, then typically these enzymes are upregulated. This ability to excrete the ammonia from the amino acids gives also the opportunity for kidneys to generate the carbon skeleton, and this carbon skeleton can be subsequently used in the gluconeogenesis. And therefore the kidneys, that's the second organ in the gluconeogenesis, together with kidneys and with enterocytes, all three these tissues are able to get rid of ammonia. Liver cells incorporate it into urea, and enterocytes or kidneys can excrete ammonia once in stool, once in the urine, and the carbon skeleton can be used for the production of glucose, and subsequently by glucose 6-phosphatase, the pure glucose excreted into the bloodstream. So, any question to this?
You can just repeat the last part of the gluconeogenesis.
The thing is that typically the tissues that can do intensive gluconeogenesis, you know that they have the glucose 6-phosphatase, and they have effective mechanism how to get rid of the ammonia. For the majority of gluconeogenesis, if we do not count the Cori cycle, it's from amino acids. So it's quite useful if the tissue itself is able to get rid of the ammonia, and then the carbon skeleton can transform to the other tissues. Therefore, this is very useful to combine these two properties of the tissues. That's everything.